Hello, my name is Kareem El Rais, and this presentation is in fulfillment of Understanding the Brain, the Neurobiology of Everyday course on Coursera. And I'll be talking today about how caffeine works. Let's talk about the different components that affects our brains and makes us sleepy or awake and how that works, the whole mechanism. Simply, there is a chemical, a neurotransmitter in our brain. It's called adenosine, and it's a byproduct of the breaking of ATPs and cyclic AMPs, which is the main source of energy in the human cell. This byproduct, when its level increases inside the cell, it gets released to the extracellular space, to the outside of the cell, with the means of transporters. This substance, adenosine, when it's released at an increased rate, it binds with two neuroreceptors in the brain. One of them is called A1 and the other one is A2A. And when they bind with them, that decreases the wakefulness and increases the sleep pressure in the basal forebrain. Now, the caffeine, which is a psychoactive drug, is an adenosine antagonist. And antagonist in neuroscience means it blocks the adenosine or the neuroreceptor from binding to its receptors. Binding the neurotransmitter from binding to its neuroreceptors. So it antagonizes it. It takes over its place, prevents it from functioning with its designated receptor. So that's what's meant by an antagonist. And that's what caffeine exactly does. It blocks adenosine from binding with these two neuroreceptors and hence increasing wakefulness. Also, something else, adenosine interrupts binding the dopamine to its receptors while caffeine doesn't. And that's why when we drink caffeine, when we drink coffee, tea, or any drink that has high content of caffeine, we have a sense of pleasure and joy. While adenosine can do the opposite because we would feel a little bit down when we get tired, when we feel sleepy because of the decreased amount of dopamine. The adenosine, as we said, binds to two neuroreceptors. The A1, which is an inhibitory receptor by nature, and you can find it in the cortex, the hippocampus, the thalamus, and the cerebellum. As we can see here, hippocampus here, and we have uh, the thalamus here as well. We have the basal forebrain, we'll talk about it. While the A2A receptor is excitatory by nature and it's expressed in the striatum in the basal ganglia. And these are the places where most of these receptors exist. However, they are in other places in the brain, but that's not in the scope of the presentation. And basically, the idea of blocking is that caffeine, or imagine the neuroreceptor, is like a holder that accepts a specific shape. Okay, and because caffeine and adenosine, they have similar chemical structure, so caffeine can sit into this holder, and that means that the adenosine, when it comes in, it can find a spot for it to sit in, which is this holder. That's the neuroreceptor, as we see here, and that's how it's been blocked. We can see here how the chemical structure of the caffeine and adenosine have some similarity, and how that helps in for the caffeine to bind with the A1 and the A2A receptors. And that takes us to something else. It will never work, or it will not always work the same way. Like if we drink coffee every day, yes, it will keep us awake for some time. But at a certain point, the number of neuroreceptors needed will increase. The human body, or let's look at the cells, if it has, a number of neuroreceptors. When we drink a lot of coffee or a lot of caffeine that binds with all the receptors, the brain will start to create more receptors of those. And that means that we'll develop tolerance to the caffeine. We'll need more caffeine to stay awake. And that's how people, they start with one cup of coffee, then two, then three, to keep themselves awake. That's because the number of adenosine receptors, or the A1 and A2A receptors, increase. However, if we stop drinking coffee, these receptors will start to disappear, and only the original ones, or a specific number of the adenosine receptors, will remain. And that also is accompanied with the withdrawal symptoms. These are the references for this presentation. You find it in the last two slides. I would like to thank Coursera, the team working on the course, and Professor Peggy Mason. Thank you.